Thank you very much. Today I'm going to tell you a story about cancer drug discovery and about trying to build molecules that target proteins that drive the human cancer. And the protein that I'm going to talk about today is called signal transducer inactivator of transcription 3 protein, and it's shown here. Now, I didn't always do cancer research. In fact, when I started, I was a synthetic organic chemist, and I'll just give you a little bio. And I started making molecules really to make the hardest molecule that I could. And for me, that was interesting because I wanted to do something that nobody else had done before. So I did this at the University of Glasgow, my PhD. This is not to be confused with Hogwarts, because it does look like Hogwarts. Interestingly, my undergraduates and my student evaluations always say that I look like Harry Potter. But I really don't think I do. So I grew a beard to try and stop these comparisons, and that didn't work out too well. Moving on, I went to Yale University for my postdoc, and at Yale University, I discovered a new field of research, medicinal chemistry. And the molecules that I made had a purpose, and that purpose was to actually kill selectively cancer cells over healthy cells. And for us, and for me, that was transformational. I really wanted to do this as part of my future career. It was hugely rewarding. So today I'm going to tell you about what we did. What we did at Yale was target STAT3, and this is the first time that I was introduced to this protein. And I found this protein really interesting because it was termed a master regulator of the cancer cell that drove the processes which made the cancer cell behave the way that it does to form tumors. And for me, this was something that I wanted to do. So I'm going to tell you a little bit about STAT3. As you can probably tell, I'm Scottish. I've been here for 10 years, but my accent hasn't changed very much. And I'm from a little island on the west coast of Scotland called Bute. And if you look at the shape of Bute, it looks almost identical to STAT3. So maybe... Maybe it was fate that I was meant to target this protein. So I'm going to tell you a story about making drugs today. Legal ones, not like Breaking Bad, but what I can tell you is that if U of T students decided to make crack, they could make it way better than Heisenberg. But of course, they wouldn't do that. Now, I'm going to tell you about STAT3. So STAT3, master regulator of cancer, drives the cancer cell processes. And what's most interesting about this protein in the cancer cell is that if we knock it out, we find that the cancer cell actually commits suicide. So drugs targeting this protein can make the cancer cells commit suicide and die. So this seems like a really appealing target. So why do drugs work in the human body? Well, to answer this, you have to think of yourself at the highest echelons of educated society. You have to think of yourself as a chemist, an organic chemist. And organic chemists see the human body in terms of a large reaction chamber. We see it as a collection of large and small molecules that are undergoing chemical reactions, complexation events, to mediate the processes, the biological processes that allow us to function as human beings. For example, serotonin, this small neurotransmitter found in the brain and in the GI tract, binds to receptors to give some of our functions today, our levels of anxiety, our levels of depression, depend on this interaction with receptors. The key point is that drugs interact with molecules, and they can do this positively or negatively to regulate biological processes and to elicit biological responses in a human. So what is medicinal chemistry? At its most fundamental level, it's the design and synthesis of molecules that bind to a target receptor, typically a protein, with exquisite selectivity and potency. And the result of this complex is a biological response. And in cancer research, that biological response has to be death. We want to see our drug binding to our target and leading to the cancer cell dying. So why does it need to be potent? It has to be potent because we want to use very little of it. Why does it need to be selective? Because our target isn't the only protein that is in the cancer cell or the human body. There's lots of different targets. So to give you a sense of the numbers, and this is pretty baffling when you think about it, the average drug dose gives the human body 10 to the 18 molecules. So that's a lot of zeros. There is 10 to the 14 cells in the typical human being. Each cell contains 10 to the 10 molecules. So if you do the math, it's a pretty amazing number that for your drug to find its target, it is a 1 in a million chance. So it tells you that when you get into the cell, you want to be binding to your protein for long enough such that you elicit that biological response before it disappears. So where do we target? So our idea was to target STAT3, but the active form of STAT3 is actually a protein dimer. And what we wanted to do was to make molecules that could come in and break open this dimer. And the dimer is the active form of this protein. So how did we do this? Well, we took inspiration from nature, and we used nature to guide us in our drug discovery process. And what you can see here is in yellow, you've got this protein complex, and this is actually DNA bound in the middle. You can see that this dimer is mediated by interaction with the yellow parts of the protein, which are shown to bind to the protein. And so our idea was, can we take this small sequence and use it to inspire the design of small molecules which will bind with higher potency and selectivity than STAT3 itself and disrupt this active form. So medicinal chemistry is a little bit like a jigsaw puzzle. We're trying to fit the molecular pieces into the active site so that it binds potently and selectively. These are two words that I'm going to be using a lot in this talk. So if we look at the active site, there's 3 main pockets within this protein. So we want to make molecules that bind to all 3 of those pockets. So this is a molecule that came up through a computational screen. We're going to call it compound 1. And it's got an IC50 of 90 micromolar. So what's an IC50? An IC50 is the concentration of drug that's required to kill 50% of the cells. So micromolar suggests that it's very small. If you remember your organic chemistry, you'll know that this is a very small amount of material. But in fact, that's quite a lot when we consider what it takes to be a drug. To be a drug, you need to have a number closer to 0.5, 200 times more potent. So you need 200 times less drug to kill the same amount of cancer cells. So what did we do? Well, we decided that to do this properly, we needed to find a piece that could fit in that top pocket so that we're tailoring the molecule for that particular protein. So we built the molecule. We added to our structure so that we're fitting into this top part of the pocket. And you can see that it's kind of like a jigsaw puzzle. So what do we do? What is the process for making molecules that kill cancer cells? How do we rank them and assess them? Well, first of all, we have to make them. And we make them with organic chemistry, which is everyone's favorite subject at university. I know my students love me, and they love being taught organic chemistry. Biologists call us cooks. They call us cooks because we mix things together and something comes out at the end. They obviously have an inferiority complex. And this inferiority complex stems from the fact that chemists are able to manipulate material at the atomic level. We can do so many things with molecules. We can even time travel. As in, we can make old men excited. We're looking out for ourselves. So the first step is to make molecules. So here's a typical synthetic scheme. We don't just make one molecule. We make, on average, maybe 25 to 50 molecules in the first pass. And we're just going to denote them as little shapes here. So what's the next step? The next step is to test whether it binds to the protein. So we do this outside of the cell. Because if it doesn't bind outside of the cell, it's not going to bind inside the cell. So the idea is that we're looking for a molecule that sticks with high affinity to this protein. What's the next stage? The next stage is to see whether it is selective. And we do this by screening against different proteins that are both similar and different. And what we want to see is that the molecule does not bind to the protein. It's not a promiscuous drug. So then the next step is we put it into cells. So we put it into cancer cells, we dose the cancer cells, and what are we looking for? We're looking to see that the drug does what it's supposed to do, which is to actually kill the cells. If it kills the cells, the molecule moves forward. What's the next stage? Selectivity. Does it kill normal cells? Do we see a selectivity of effect for cancer relative to healthy cells? And ultimately, what we want to see is no effect whatsoever. So the next stage is we have to do some cell biology. We have to look inside the cell and see whether we're knocking out our protein target. So what we want to see is with increasing concentrations of drug that we see a decreasing amount of our protein. And you can see it here. Then what do? We look at other targets inside the cancer cell. We want to see, is it hitting everything? And the goal will be to see that actually nothing happens to other cells unless they're related to our protein. We want to see no effect. We want to see an effect on our target. So the final stage is looking in vivo. So in cancer research, we engraft mice with human tumors, and we dose the mice with our drug with two objectives. The first objective is to see whether or not the drug is efficacious. Does it shrink the tumor in the mouse? And the other thing is it toxic? We're going into a living system. We want to see whether or not we have serious side effects as a result of this drug. So what did we do? So we started off with this molecule, compound 1. It's got an IC50 of 90, and remember, what we're trying to do is to make that number significantly smaller. We want to see this number dropping way down. So at U of T, we have a strict tenure process, so people like me have to do well, not just in one project, but in multiple projects, or else we get fired. So bearing that in mind, we made our first change, and our first change was to get rid of this oxygen, replace it with a nitrogen. And that turned out horribly. So at this point, I was thinking of career choices in Scotland because Canada was going to kick me out. But remember the policy that we had, that we wanted to access this upper pocket of the STAT protein. So we put in a benzyl group, 300. Paracyano, 260, terbutyl, 190, biphenyl, 115, cyclohexylbenzyl, 4 micromolar. So this compound is now 20 times more active than the first molecule. So that was really exciting. We were very happy, but it wasn't good enough. We wanted to be into the 0.5 region in terms of going forward into animals. So we made some more changes, and I'll summarize it very briefly by saying that we discovered this molecule, BP1102. So we changed this part of the inhibitor. And so this molecule was way more potent. It was 0.8 micromolar. So we were getting more and more potent. We were using less drug to kill the same number of cancer cells. So what did it do in cells? So in healthy cells, we found that it had no STAT3. We saw that it had no effect really on them at all. When we put them into cells that had STAT3 and were tumor cells, we saw that it killed the cells really, really nicely. And so then we decided to move into cell biology. And we saw, with increasing concentrations of drug, that our molecule decreased the amount of STAT3. So it showed us that we were on target. We were selective for cancer cells over healthy cells. So now what did it do in an animal? So the first thing we do is we test in a mouse. So we inject it into the mouse and we look to see how much drug is there and for how long. So the interesting thing about animals and humans is that they have a liver. And a liver is responsible for trashing most drug discovery dreams because the liver is full of enzymes that metabolize drugs. So think of the analogy of the jigsaw puzzle and think about all the time that my poor graduate students put in to identifying this molecule. And then they inject it into mice, and guess what happens? It completely changes. The entire structure is different. And that sucks because it doesn't fit in the protein anymore. And we have to go back to the drawing board. To give you an idea of how efficient the liver is, this drug is gone within 10 minutes in the animal. Completely gone. There's nothing left. So what do? Like so many things, we rely on chemistry. And we rely on chemistry to stabilize these parts of the molecule that are considered weak to metabolic enzymes. So, to cut a long story short, we ended up making this molecule, which is a derivative of BP1102, and we saw that this molecule was way more stable. In fact, it has a half-life of over 4 hours. So this was really exciting. And then we did some more chemistry, but it's patent protected, so we can't show you the structure, but we'll call it Compound X for my venture capitalists. So we went into a multiple myeloma trial. So multiple myeloma is 100% lethal disease. It's a really toxic cancer that claims everyone who gets it. So right now there is no cure for it. So I'm going to show you the effect of our drug in multiple myeloma, and I've shown you a series of images here which are really cool, and I'll just state that again, that these are really cool images, because your idea of cool and mine might be quite different. So what you're seeing here in these mice is actually a tumor, but you're also seeing STAT3. So STAT3 activity is labeled. So you see this fluorescent dye, it's associated with STAT function. So what we did was we treated with Compound X, and what we showed in a time-dependent manner over an 8-hour period was that our molecule shut down STAT3 inside a human tumor inside a mouse model, which we thought was really cool. We then went into a medulloblastoma trial. Medulloblastoma being one of the most common brain cancers in children. 70% of the patients are treated with really a horrible cocktail of drugs and radiation, survive. They're left with severe side effects that they'll take with them their entire life. The other 30 to 35% are palliative. There is no cure option for them. The tumor spreads down the spinal column. So we wanted to see whether our STAT3 drug would have an effect on this recurrent medulloblastoma, the medulloblastoma that is completely untreatable right now. And what I'll show you is two things. Here is brains slices taken from mice that have got these brain tumors, and you can see the tumors in purple. And then we treat with compound X, and what we saw was really stunning regression of an untreatable tumor. So this clearly got us very excited. And just to show you that it actually has a difference on the quality of life of these mice that have human tumors. This is a wee video, and what you can see here is a mouse that has been treated with compound X and a mouse that has no drug, and it just shows you the mouse that has treated with our drug is quite happy and behaving like a mouse does, energetic and inquisitive, whereas the mouse that has had no drug is clearly moribund. So finally, to summarize, I hope I've shown you in the course of this talk the rational approach that med chemists take to discover new molecules. And we've gone from a really horrible compound to what we think is a really good compound for treating untreatable orphan diseases right now, compound X. This compound has just received Series A funding, so we're hopeful that we'll be in clinical trials now within the next 24 months. And I will add the caveat that it is barring hiccups. Medicinal chemistry and drug discovery throws up hiccups all the time, and hopefully we don't experience any more. And finally, I'd just like to acknowledge my research group. These guys do all the work. They're the ones that are making the molecules, testing them, and I just have the pleasure of telling you about it, and thank you for your attention. Cheers.